Okay, so in this video, we're going to look at how scientists map the history of life on Earth using phylogenetic trees. So when we look at what a phylogenetic tree shows, it shows the evolutionary history of species and their relationships and how far back in time they had common ancestors. So if we look at like these horse-related species, we can see a couple different ways of like demonstrating the evolution of horses and zebras and donkeys. You can see here in this one, it's more like an actual tree-looking shape. This one you have more like rectangle pattern, and you can present it this way. So phylogenetic trees, basically what we are looking at when we see a tree like this is we are seeing like the different speciation events over time in different species, and it basically shows common ancestry and how related they are. So there's two different ways that trees can be drawn. We can draw them with like this rectangle shape or like the Y shape kind of. And so regardless though, is when we look at a phylogenetic tree, at the bottom represents, think of it like the roots of a tree. So that is the evolutionary past, which could be measured in millions of years, hundreds of thousands of years. It really just depends on the time scale that is in the context of that species. But when you go from the bottom up, that is from the past to the more recent. Here I have it as the present, but if you're looking at like dinosaurs or something that went extinct so many millions of years ago, it would just be like the most recent at the top. But I also want to show you that when you look at them sideways, so on this one that has like right here, this would be the past, and then as you move to the right, that is your more recent or the present. And then if you have one that ends early, like this one right here, that is an extinction event, like that species went extinct. Okay, so now let's go ahead and see how you read these and what they mean. So at this point right here, where you have this like branching event, that represents speciation. That represents a time in its evolutionary past where there was a common ancestor. Like think about one population that then became separated. So it could have been due to like geographic isolation. And in that speciation event, you now have allopatric speciation, and now you have two separate species that no longer have gene flow between them. They are reproductively isolated, and now they're on their own evolutionary like trajectories. However, it could also be sympatric speciation and have it speciation occur while living in the same place. It doesn't geographic region. It doesn't have to be geographic isolation. Really, the key here is that they have become reproductively isolated, and at this moment in time in history, that is the last time they shared a common ancestor. So when you look at like this here, you would put this is the most recent, and this box right here represents the common ancestor of 1, 2, 3, and 4. So if I were to ask like where on this tree is the last time 1 and 3 shared a common ancestor, it would be here. If I asked where's the last time 2 and 3 shared a common ancestor, it would be here. The last time 3 and 4 is here. Okay. So we also want to talk about when we look at the evolutionary history of a species, like here you have species A and you have species B. With this blue line represents their shared history. So any like mutations or behaviors or phenotypes or genotypes that evolved in that common ancestor will be in both species A and B at this point in present day, because this is like a shared evolutionary history. And then at this point here, they split. And yeah. Now, if we were to talk about, though, let's pretend it's something like a reptile, and you have like a common ancestor that lays eggs, has scales, has 4 legs, adapted to living in dry, hot environments. So you have, let's pretend it's the common ancestor to snakes and lizards. Well, that common ancestor may have had 4 legs. It's totally okay, like in species line A right here, if that were snakes, to say like at this point or in A's unique history, it through natural selection lost their legs. Like sometimes you could have a shared trait that ends up being selected against in one line, and therefore you can also lose traits evolutionarily, not just gain traits. Okay. So if we were to look at penguins as our example, because I think it's easy to picture with movies like Happy Feet and stuff. So if we have an ancestral penguin species, like here is one I got from Google Images, but let's pretend right here represents the evolutionary past of these penguins. And at this point here, you have a speciation event where maybe we have geographic isolation where the penguin populations were separated. So this would be the most recent common ancestor. However, once you have these lines separated, they represent time. So as you go through time, like from the past to the more recent and to the present, you have mutations, genetic drift, you have natural selection, you have evolution happening along that time. And so in that evolution happening, those changes in the population of these like spotted penguins is unique and different than the evolution that's happening along this time. And so we need to remember that these two groups are geographically or they're reproductively isolated and they are not having gene flow. There is no mating occurring between them. And so their own histories are now separate. I also want to point out that when we look at these species as you move through time, here, like this penguin that I have right here, maybe is what the common ancestor looked like at this point. So from this penguin with the long, long beak, way back, I don't know, however many hundreds or thousands or millions of years ago, I don't know the time scale for penguin evolution, but let's pretend right here is what it looked like. Well, now in this line, it has its own evolution happening. Maybe it's gradualism that's occurring and it's slowly changing over time. And then at this blue line, it now has evolved to be this species. So whatever the common ancestor was at this point, it's different than what it was millions of years or hundreds of thousands of years in the past. It has its own evolution occurring before the split. And so to go from this common ancestor to present day, maybe along the way there was a penguin that looked like this, kind of showing the transition from the recent common ancestor to the present. So in the fossil record, we can actually see transitional fossils that show us these changes over time. And maybe in this other group, maybe like a spots was a mutation that showed up and it ended up being selected for. So you have in this evolutionary line, penguins with spots. You can also see this beak type is different, and so maybe a more round beak was selected for. Now, if we wanted to show another speciation event, we would just simply add another line that we can see right here. So now these two penguins have a really recent common ancestor right about here. Okay, so at this point marked with an arrow would have been a speciation event or a reproductive isolation. Now, when we look at how you read these phylogenetic trees and you talk about shared derived characters or shared derived traits, what we can see here is that polka dots is a trait that's in a shared history between A, B, and C. And so polka dots would be in all 3 of these species. But 3 claws, which these are all things I made up, would only show up in species A. It is unique to species A, where a curly tail is in the common ancestor of B and C. So you'd find a curly tail in B and C, and rainbow looks like was selected for in the unique history of C. Okay. So when we look at these penguins, these circled areas, not only do they represent like speciation, but they also represent the last most recent common ancestor. So if I were to think about like where on this phylogenetic tree is the common ancestor between 6 and 7, it would be part B right here. And then for 6 and 4, A would be the common ancestor. All right. Okay. There's a worksheet you can do in class. Now, are all of these trees showing the same thing? Yes, they are. Okay. So now, when we think about these trees, though, like where do scientists even get the information to know their relationships, right? So we can look at like two major sources. Is we have our fossil record is especially important for your species that are extinct because extinct species, we may not have access to their amino acid sequences or to their DNA, but we do have access to their structure, their morphology. So we can use the morphological data to draw our trees. But if we have living species, it's going to be more accurate to use molecular data. So morphological is good if that's the best you have. But in morphological data, you do need to be aware that things like convergent evolution can be misleading. So if you look at like a mole, like a burrowing animal underground, and you look at their fossils in North America, and you find a burrowing animal in Australia, and you look at just their fossils, they are going to look very similar. However, it's not due to common ancestry, rather because they both burrow underground, and by natural selection, certain phenotypes were favored. So that, like similar morphology, would be incorrect to connect those as if having a recent common ancestor. Now, your molecular data is actually going to be your strongest because when you look at like evolutionary time, we have these things called molecular clocks where you can like kind of, there's like a certain rate that mutations happen at. And so you can make inferences based on the number of differences in DNA, like how long two species have been separated for. So for example, if I were to look at this first penguin, we'll call it penguin 1 right here, and penguin 5 down here, you can see their most recent common ancestor is kind of way back in time. So with those two penguins, they would have, like, if you were to count up their DNA differences, like how many mutations have made them different over time, I don't know, we might find 15 differences versus penguin 1 and 2 may only have 3 differences because they've been separated for less time. Therefore, mutations that happen in a common ancestor will be in both. And it's not until they start to split and diverge that now mutations are showing up in their own individual lines, and that shows, oh, there's differences in their DNA. But if they only have 3 or 4, 1, 2 differences, that means they haven't been separated as two species for very long. But if you have 15, 16, 17 differences in a particular gene, evolutionarily your lines diverged a long time ago, and there's been quite a bit of time in order to have mutations occur between your two groups. Okay, so when we look at our species, like here, you can see this is called the genus, and then you have the species. We use a binomial nomenclature in order to name our different organisms on Earth. So we have like our taxonomy or our classification system. Now, what's not pictured here is something called domains. When we look at all life on Earth, we can group them in 3 separate domains. You have the eukarya, which would include all eukaryotes. Then we have our like archaeans, and we have our prokaryotes, or bacteria. Or not prokaryotes, sorry, bacteria. So now if we go with eukaryotes, in our eukaryotes, we have different kingdoms. We have plants, animals, protists, fungus. So here we're looking at our kingdom Animalia, which would include all animals on Earth. We want to be more specific though, and so now we're going to go to the phylum Chordata, which is all things with a backbone. So jellyfish would not be part of this, or insects or invertebrates. Then we get more specific because we have reptiles, amphibians, fish. So now we're going to go to class Mammalia, order Carnivora, family Ursidae, and genus Ursus, and then species. So this is how we get more and more specific to be able to identify a species on Earth. All right, this is part one. Okay, good job.